My research focused on the neuroprotective effects of adequate folic acid intake on overall DNA methylation in a murine model of Alzheimer's disease. I did this research in conjunction with my lab partner, Aubrey Beaver, and my mentor, Dr. Susan Helm. First, I think it's very important for this research to have a basic grasp of the folic acid metabolism cycle. Two important things to note on here that I will reference throughout this presentation are folic acid versus folate, and just noting that they enter the pathway at different cycles or different points in the cycle. I just think this is important to understand that dietary folate and folic acid supplementation are two different things. We do not supplement with folate, we supplement with folic acid. Folate can only be consumed through a diet, so just noting that they enter the cycle at two different points is important. This does not necessarily affect the results in terms of recommending if somebody were to supplement or consume folate directly from their diet. We reach the same conclusion in the end. The other important thing to note for this specific research is on the left, the DNA methylation kind of side pathway. We look at hyperhomocystinemia, and this is something that we see a lot with Alzheimer's disease. So this would be an excess of homocysteine in the blood. And when we have a significant amount of homocysteine, that likely means that less homocysteine is being converted to methionine. Therefore, we have less of that 5-methyl group byproduct, and that is what we use to methylate our DNA through another pathway cascade. So when we have too much homocysteine, we don't have enough methionine being made, and therefore we don't have the methyl groups in order to methylate our DNA. So we would expect to see a lower amount of methylation when there is hyperhomocystinemia. We suspect, or has been hypothesized, that homocysteine can affect the amyloid beta fibrinogen interaction. And without getting too much into it, amyloid beta is very key in the pathogenesis of Alzheimer's disease. So potentially determining that pathway would be very important in implicating hyperhomocystinemia as a potential etiology to some of the pathogenesis of Alzheimer's disease. We basically suspected that an increase in the folic acid that we were supplying in the diet would allow for more homocysteine to be converted to methionine, because we have an increase of 5-methyl tetrahydrofolate at the end of that folic acid metabolism cycle, and more of that would yield more of homocysteine being converted to methionine. So our thought process was, by increasing folic acid, by increasing 5-methyl tetrahydrofolate, will decrease the amount of homocysteine, increase the amount of that being turned into methionine, therefore increasing the amount of methyl group that we have available for DNA methylation. Our research question, I basically just stated it: will dietary folic acid supplementation facilitate a greater effect on DNA methylation in an Alzheimer's disease population compared to a healthy population? We pose this question simply because we suspect that the Alzheimer's disease population has impaired DNA methylation compared to a healthy population because of the pathway that I just discussed. For our methods, we got C57 black mice, just a standard laboratory mouse. They had 5 weeks of habituation, just getting used to a new environment, new routine, and then they had 8 weeks of experimentation. So these were randomly divided into 2 groups, and then they lived within those groups in little groups of 4. This was just so it was easy for us to identify mice, keep any disease contained, as well as their social creatures, so we didn't want them living completely alone. They had ad libitum access to food and water, so there weren't scheduled feeding times or anything like that. They could eat whenever they wanted. The diets, which I'll get into on the next slide, did contain certain compounds that were just there to inhibit folic production by intestinal flora. So we just wanted to make sure that the only folate they were getting was from their diet or the supplementation, to really make sure that our results were true to what we were testing. And the folate amounts in the diet we determined using the recommended daily allowance for rodents, which is set by the American Institute of Nutrition. So the standard diet is just going to be 2 milligrams of folic acid per kilogram. All right, so our control mice and our Alzheimer's mice, the controls were injected with saline every other week just to keep their treatment pretty much the same as the Alzheimer's group. The Alzheimer's group was injected with DSP-4. It's a neurotoxin that we used to simulate Alzheimer's disease. It's relatively commonly used in this line of research. And each group had an N of 75. So we had that total N of 150. And then you can see on the bottom in the diagram that those were then divided into 3 subgroups each: folate deficient, standard, and folate excess diet. Each of those had an N of 25. To test the DNA methylation, the mice were sacrificed at the end of the 8 weeks, and we took a lot of different tissues from them. I only tested the methylation in their liver tissue, and I used the 5MC DNA ELISA kit from Zymo Research. It had a horseradish peroxidase developer, so that bound to the methyl groups essentially, and the more absorbance that was read in the ELISA plate reader, the more methylation that there was, and the less absorbance that was read, the less methylation. Our absorbance was read at 405 to 450 nanometers. And then we performed a one-way ANOVA to identify group differences across all 6 groups. These are our results. The control group is on the left, the Alzheimer's group is on the right, and the scale is the same across both graphs, so we can directly compare them. As we can see, we were reaffirmed in the fact that the methylation was lower across the board for the Alzheimer's group compared to the control group. So, like we were thinking in the beginning, that hyperhomocystinemia that we see with Alzheimer's disease did yield that lower methyl group availability and lower methylation overall. We're going to mainly focus on the control diet versus the folic acid deficient diets to compare a population that was deficient in folic acid versus a population that had adequate folic acid. We included the high folic acid groups in order to see if there were any detrimental effects or opposite effects from kind of reaching that upper limit of folic acid recommended intake. But for the purpose of these results, we're just going to focus on the control and the deficient groups. So we saw opposite effects in both of the groups, which was a little bit surprising. The control group, from the control diet to the deficient diet, we saw a decrease in methylation of approximately 49%. And in the Alzheimer's group, from the control diet to the deficient diet, we saw an increase of approximately 45% in DNA methylation. These differences were significant. Our ANOVA had a p-value of less than 0.05. And essentially what this means is that we have an altered DNA methylation pathway in Alzheimer's disease. We can kind of take this hyperhomocystinemia idea a step further and say that it likely is causing some alteration in DNA methylation. An increase in DNA methylation we would think would mean a decrease in overall gene expression. In terms of how that affects Alzheimer's disease, that was beyond the scope of this study and beyond what I know in my education. I'm not a geneticist, but that would be something that we would want to explore in further research. So how does then this result, what does that mean for gene expression in Alzheimer's disease or other neurodegenerative diseases? And we do think that folic acid supplementation may slow the progression of some of the Alzheimer's disease symptoms by this pathway. If we can kind of normalize those homocysteine levels, if we can normalize the interaction potentially between amyloid beta and fibrinogen, and if we can kind of offset the hyperhomocystinemia, could we potentially alleviate some of these symptoms of Alzheimer's disease or slow the progression of Alzheimer's disease or just specific symptoms? So, in conclusion, we believe that further research into the potential neuroprotective effect of folic acid supplementation in Alzheimer's disease, as well as in other neurodegenerative diseases, is warranted. Our next step was to look at Down syndrome because Alzheimer's disease has such a high incidence in the Down syndrome population, and we often see similar etiologies in terms of brain function and things like that. We wanted to explore that pathway and see what other kinds of neurodegenerative diseases might follow a similar pathology that we saw here. Thank you.